All right, everybody. I'm coming to you from Boston today, and I'm running around to different meetings, so that's why. As Sergey pointed out, looking a little fancier today. Thankfully, this Starbucks has good Wi-Fi that I'm in, so I'm sort of squeezing this in the midst of some running around. I think we may have a few more joining us in a little bit, but we'll go ahead and get started. We've got somebody new here joining us, Gopal, who's a new volunteer, who's been looking at some of the muscle model and high OpenWorm stuff. Hi, Gopal.
Hi, everyone.
I know you don't have video going, but do you want to just say a little bit about yourself and like where you're located geographically and how you found OpenWorm, what you're interested in?
Sure. It's my background is in math and physics. I'm working as a software engineer now, and I'm in Boston.
Oh, you're in Boston too.
I am in Boston, yeah.
I missed that point. We should get together.
Definitely, yeah. Send me a line.
Yeah, I'm here for a meeting in like biology and technology. Sure enough.
Okay.
Right now I'm in Somerville, but I'll be mostly downtown. Cool. And yeah, so you linked up with us a couple months ago, I think.
Yeah, that's right. How did I find out about OpenWorm? I'm not even sure. I think I might have Googled simulating CL games or something like that. And so yeah, I was pretty excited to find out about you guys, and haven't really made too many inroads in yet, but I'm hoping the next couple months to get things off the ground. I guess I should say that I'm starting medical school in a couple months, so
Okay.
I'm hoping to, yeah, kind of have this perspective of what it takes to do a realistic biological simulation. As I pour through 8 hours a day of learning anatomy and physiology and biochemistry and things.
Awesome. Yeah. Well, maybe we can even catch up this evening. I don't know what your plans are, but
Yeah, let's try to figure something out.
okay. I can send you some mails offline. But
Sounds good.
so in the room here, there's Andre and Sergey, who are both in Novosibirsk, and they've built out the mechanical model of the worm that we call cybernetic, or smooth particle hydrodynamics. And Giovanni there is in Oxford, and he's sort of been heading up the whole worm sim effort to get all this stuff packaged up into a web form and pushed out amongst other things. And we're all sort of co-founders. I've been with the project from the beginning, and then I do various coordination stuff, and allegedly I do some science from time to time, but there's a lot going on. So welcome.
Thank you. Great to meet everyone.
So the way we've been structuring this meeting, and again, there's several others who hopefully will drop in, but we'll see. The way we structure this meeting, we just kind of do a round robin usually to get updates from folks and see what folks have been doing. I usually start with some organizational updates, but then we also have started a thing where we do kind of a bit of a deeper look, like a 15-20 minute presentation in one area of the project every month that we do the meeting. And so Andre will be doing that in a minute. And but first, let me update some things on the organizational side. So you should have the link to this, be able to edit it. It's publicly available. Okay, so a couple of things, guys. So Google Summer of Code via the INCF has let us know that we will have a student assigned to OpenWorm reports. We're not allowed to say who that student is yet until after the 27th of April. So I can't report it here, but we will have somebody that will be working with us through that program. So that's great. Last year we had 2, and this year I think the slots were a little bit more constrained, so we were very pleased to get 1 student. So look forward to seeing an announcement on that coming out by the 27th. But that always adds some good energy to have somebody who will for sure be able to devote like large chunks of time over the summer. So that's the first thing. Second thing, So you guys remember, like way back at the beginning of the project, we got a Amazon Web Services research grant, and that let us use Amazon's cloud servers for a while, and we were very grateful for that. We reapplied the next year and tried to get that again, and we heard like a resounding silence from Amazon Web Services for like a year, and I kept like trying to ping them back and nothing. Well, they've resumed the conversation with us finally through another contact that I have, and they're talking with us again. They found our old application, and they're now processing it again. So I've been having some talks with them about what we could use that for. I'm hoping that two things that come to mind are pretty obvious: is like help us to host WormSim, and also to help us to host the videos for the movement validation group that we have up on our terabyte server. Are two things that folks have requested. So we'll be talking more with them about sort of credits for those things. But that has sort of, to some extent, slowed down the plan to put the videos up online directly from the server, although we may still do that in parallel. But worth having the conversation with them to see kind of what they'll do from that forward. So thank you, Amazon Web Services, for talking to us again. Hopefully we can get some cool new space and maybe use some of their high compute clusters to speed some things up. I don't know. There's a lot of different possibilities. Should be good. I also got to present, I guess I didn't write here, but I also got to present OpenWorm at a Future of Engineering conference, like a bunch of software engineers over the last weekend, and got a lot of other interest in the project from that. So I will let you guys know about any additional like contacts that come out of that are actually useful. But basically, it's like companies that make computer-aided drafting software, like Dassault and Autodesk, are talking to us now a little bit. So we'll see if there's additional interest that comes out of that. But it's kind of cool. So that's kind of what's been going on with me. I should also say that in the realm of the like high OpenWorm muscle model stuff, this has not been a great month since we last met for me. So there have been less communications coming out of me, and I'm being a bit of a bottleneck with some of those things. But this month that follows, hopefully should open up a bit more for some of those things. So I will be, if folks are watching and haven't heard from me, this is why. It's been an unusually busy month for some of the other stuff I'm doing, so I haven't been able to push some of those things forward, which of course happens. We all get busy in this community, but the following month should be easier. And Gopal, since that's a topic especially that you're interested in, this will also be a good conversation for us, as well as we're getting Google Summer Code students that are in. It'll also be good to kind of move those pieces forward. So it hasn't. The effort hasn't died. We're going to continue it, but sometimes it's a little harder to put in the effort when all these other things are going on. So that's my basic update. So from here, let's go ahead and go on to Andre, who will tell us about the new stuff that he's working on in some length, and hopefully show us some cool pictures. I'm hoping.
Okay.
Okay.
Possibly I will try to share my screen.
Great.
Maybe.
I should also say the other thing that's been going on this month is that Andre, Sergey, and myself have been working on revisions to the SPH paper. We've had like 2 or 3 meetings on that topic. And we're making good progress on revisions.
Okay, it seems visible, right?
Yep, I can see it.
Great. So I will try to speak about the real complexity of C. elegans nervous system, about things which work, about mechanisms which are beyond the connectome, something which is sometimes even more complex than we can think of it. So first statement, people often say and even write in papers that C. elegans map of connection between neurons or connectome is completely determined, or connections are fully mapped. In reality, there is a good citation from authors who worked with the most recent version of Connectome, which is available online. In 2011 it was published, so I will just read because it's very well said. Complete wiring diagrams on the level of individual synapses remain scarce, and insights into function they can provide remain unclear. Even for C. elegans, whose neuronal network is relatively small and stereotypical from animal to animal, published wiring diagrams are neither accurate nor complete and self-consistent. Authors estimate the completeness of their version as about 90%, but it's still an estimation. Maybe it's more, maybe less, depending on the real situation. Actually, there are three alternative versions of Connectome available on the internet. Some of them even contain pictures of main neuron processes, points where synapses contact with each other. So something we can see more than just matrix of connections. But unfortunately, this data from these three sources sometimes is significantly different from each other. Okay, next. As we know, there are 302 neurons, full number of neurons in C. elegans. It is also known that 20 of them is a part of so-called pharyngeal system, which the worm uses to feed, to eat bacteria. So this system contains a muscular pump, which takes the food into itself, and it is driven by this 20 neurons. Maybe driven, maybe just influenced by, because well, it's really impressive experiment. I didn't believe myself when I read about this. People who have performed this work one by one with laser ablation technology killed all 20 neurons of this system, and the worm was still able to perform this pumping and eating bacteria without nervous system. By the way, these 20 neurons are completely isolated from other nervous system. There is some layer which is impermeable to neurons, and no neuron or its process cross this thing. This thing works with is able to work without neurons completely. So does, for example, the human heart. It also can beat without additional neuronal stimulation.
Yeah, it sort of reminds me, by the way, of how the muscles by themselves have the ability to signal each other with gap junctions and that kind of thing. So it's probably a self-contained dynamic system for pumping, even without the neurons.
Yes. In this context, it is even more impressive that in live worm, this system can be stopped. And experimentalists decide that this can be done only by means of hormonal signaling between this pumping system and the rest of the body. So other nervous system or some other organ can send hormonal signal, which maybe by diffusion or some other mechanisms can reach this pump and stop it in some cases.
Yeah, Andre.
For example, it can happen when
I have a question.
When the nematode is when it has a lot of high quality food it can eat. When it is completely full of food, it stops eating and turns into some resting phase. Also, this mode can be reached by other reasons. For example, heat stress. For example, heating for about 37 or even 40 degrees during half an hour or 15 minutes seriously damages internal mechanisms of the worm. A lot of proteins inside it become, they change their conformation or something like this. So a worm needs a few hours to fix all its damages with some inner machinery, but it needs to be in complete rest during these hours. So some mechanism stops all muscular activity in the pump, in the body wall muscles, and so on.
Hey, we've got a question for you, Andre.
Yes.
Will I ask?
Yes, go.
Yeah, sure.
So basically, I'm wondering if those 20 neurons that seem to be useless, but are they definitely motor neurons? Because they seem to be connected to the pharyngeal muscles, but are they motor neurons or are they maybe sensory related neurons?
I think all types are possible. Maybe sensory, of course, because they need somehow to sense what the worm is eating. I also read that worm distinguishes between bacteria, different types of them. Some of them are considered tasty and easy to eat because they are quite soft, and some are harder and maybe not so chemically balanced inside.
No, but yeah, I think I'm curious to know in terms of the classification of those neurons, if they're classified as, because like in the connectome, the neurons are classified as motor neurons and sensory. I'm just curious if anybody knows, but we can easily look at it on the on the worm browser, and click one of those neurons and see what it says.
Let me look while we're here.
Okay.
I'll see.
Yeah, thanks, Andy.
And of course, this 20 neurons influence pumping rate. It varies from 0.5 hertz to 4 hertz, depending on different conditions. So let's go further. When we are interested in some internal mechanisms, it seems to be a good idea to search for something which oscillates, doing some periodical activity, so we can hope to try to find the source of this activity, and there's some network which does it, but Well, as we have seen on example of pharyngeal muscles, sometimes muscles can do it themselves. Well, anyway, um, when C. elegans searches for food, its head is doing frequent movements like scanning the space from left to right, about 10 Hertz oscillations. And sometimes this mechanism can be almost immediately switched off. For example, when a worm is moving forward and then meets some obstacle, or gentle touch happens to anterior part of the worm, and it immediately switches off this foraging mechanism, this scanning with the head tip, and reverses its direction of movement, comes back, and only when again moving forward begins, it starts this mechanism again. So there should be some neuronal contour which switches this whole thing on and off depending on the situation. Additional situation when it can be switched on and off is when worm crawling on the agar surface meets a small puddle of water. It switches to swimming mode and also stops these foraging movements of the head. So here might be also quite interesting underlying neuronal mechanism, but it's still unknown. So what I'm doing, I am searching for papers and for neuronal contours which can be good candidates, potential candidates for simulation in near future. Okay, next. Also in one of the papers, I came across very interesting information. That example with crawling worm meeting a puddle on the way. Neuromediators can be responsible for switching such modes. For example, in this case, serotonin is necessary for crawl to swim transitioning, and dopamine is necessary for swim to crawl. There are mutants which lack this or that neuromediator, and in this case, they don't have ability to switch from one mode to another, or otherwise can do this but cannot do that, and so on. Also, a question arises about how C. elegans sense that there is no more ground and the puddle of water begins. It may be like we percept the difference between, for example, the air and the water of the same temperature. Water has another heat capacity, so it is sensed as cold, wet, and so on. I'm not sure about mechanisms of this kind in C. elegans, but somehow it distinguishes between water and agar surface very quickly and responds to it. Okay.
By the way, we got the answer to the question here. Put it in chat. The pharynx neurons are mostly interneurons. Some of them are classified as motor, and then a few of them are kind of dual interneuron, motor, and sensory. So.
Thank you. It's interesting.
Thank you. Okay, and a few more slides. So now about a few words about synaptic transmission. as we know, the one way is that a signal comes to the synapse. Then some neuroimmunity is injected into synaptic cleft, and then it is taken by receptors, and one way it is direct. So some ions go into the synapse and cause electric signal in postsynaptic process of neuron. And another way is indirect. It involves activating second messenger pathways in the cell, and it can cause quite significant mechanisms to run. For example, let's consider different neuroimmunity. Also impressive paper. Authors considered a number of C. elegans mutants. Each lacks one type of neuroimmunity. Surprisingly, all of them were not viable, so they can live at some larva stage, some earlier stages of development, but they cannot grow up and live during a long time. But the only one mutant which lacks acetylcholine was able to grow up until adult stage, can eat, can live. It has noticeable defects in behavior, but it's still viable. So there are a number of neurons with various neuroimmunity. So does it mean that we can switch all of them off and leave only acetylcholine neurons? It seems that I made a mistake. All these mutant worms were not lacking one of neurotransmitters. They possessed only one neurotransmitters for each mutant line or strain. So one had only acetylcholine, one had only another, second, third, and so all most common neuroimmunity. So only acetylcholine line is viable.
So, Andre, did you hang on to the references for that particularly that last statement, like the paper that says that?
Yes, for sure. I have all references. I will prepare some document and share it with all the links.
Yeah.
To these things.
That might be an interesting paper to do as a journal club, actually.
Yes. Okay, a few slides more left. One more surprise. During a long time, C. elegans neurons, which are quite simple anatomically, which are quite small in diameter, which lack sodium channels and axial potentials, were considered quite primitive, simple, and isopotential. So every cell at every moment has the same potential at all its processes within one single neuron. But 2 years ago or 3, it was found that even a single process of neuron can have compartments which have independent activity. So one can be completely off, one can be obviously on, and this activity is independent between each other and is caused by a synaptic input from other neurons, presynaptic neurons. Interestingly, this activity is coupled with head bend, left or right position. On this slide, it looks quite simple and obvious, but if we look in details on the same neurons and its points of synaptic connections, it's very hard. It contains a lot of dozens of various synaptic inputs, so not surprising that something like this happens there. But my thought is that it's very hard to simulate or even to distinguish between synaptic inputs which are important for this process described in this paper.
So there's a point here to make as well, by the way, which with this picture in particular, I think it's very interesting. You know, the idea of modeling a neuron either as a single point or modeling a neuron as multiple compartments is two different approaches. And a lot of your large-scale models will tend to just think about the neuron as a point, and certainly C302 begins with that assumption. But the C. elegans NeuroML that we've built has the multiple compartments. And this picture that you're showing right here, where you're seeing every single different connection on a single neuron that has a very complicated shape, and you know what you can do with a multi-compartmental model is move away from just isopotential, where every single part of the neuron is exactly the same value of voltage, and in different compartments can be higher and others lower. So, you know, just looking at this, it reminds me that, you know, if we want to do some interesting work of moving from single cell to from a single compartment to multiple compartment work, we could go one neuron at a time. Right now we're looking at motor neurons for things, but you know, we could pick like this neuron, and you could model every single synapse on it and the position that it's at, and you could have some interesting comparisons of what happens when information flows through the multi-compartmental version of this neuron, as opposed to what just happens with a single compartment. So I don't know, it's just some thoughts that come to mind looking at this picture, that we have like this level of, we're capable of doing this level of modeling. We have all the tools for it. We just sort of don't have as many folks that are jumping in on this. So Gopal, by the way, I'm looking at you to some extent. If you're looking for new stuff to look at, there could be some interesting things to look at, the like simulating this particular neuron with all of its connections, that kind of thing.
Okay.
So, just something to note down. Yeah.
Well, mechanical devices are quite simple, logical, and obvious for us. So there is a source of energy, there is some oscillator or engine which consumes this energy and makes something out of it, some useful work from it. It seems that living systems doesn't have some central mechanisms. Everything is shared inside them, but should we think about, or should we have a hope to find some entry point to start a simulation, something which is quite simple, works reliably, and does something which can be observed, like generation of oscillation or something like this, or otherwise, does it work as a whole thing and needs all of its components. It's very complex wired structure, full of synaptic connections, different level mechanisms, and so on. As we have seen on that example with acetylcholine, we can do not take into account quite a lot of neurons. Maybe we can do not take into account more than half of them even, and still the worm will be viable and can even grow up, eat, and crawl, and so on. So maybe this way is a good point to start. Maybe something another. Well, I tried, for example, to produce a schema of contour connected with movement locomotory system. So there is some periodical system, there is some logic. When we consider only groups of neurons like VB, for example, 1 until VB11 and so on, series of neurons of one type which are met periodically from the head to tail, one after one, and so on. But when we add to this periodical structure some other neurons, for example, that one which sense gentle touch of anterior part of the worm, the schema becomes very complex and hard to understand. It seems that there is no logic, and then it's quite hard to build a model of such thing if you don't understand how it should act, behave, and so on. So this was one attempt to do something about this. Of course, all these things can be implemented as a model in a neuron software. Maybe it will even work somehow, and it's my hope for the near future. But here we also need a discussion, good advices, discussion of strategies, of further research, and so on. Well, this is a fragment of this schema and its connections to muscular cells. Of course, not all connections. There are a lot of them, and here is only a small part.
Andre, just on that, just a question. Oh wow,
yes.
This is very cool. You've seen the papers that I think I presented some of them in the journal club a while back. That if you go back one slide, where they talk about a repeated, where they talk about like a repeated sort of schema for the nervous system. Do you know the papers that I'm talking about? They
yes.
have a. Okay, so you had a look at those when you were building this?
Yes, but I used only data from Connectome.
Okay.
So this paper, if I'm thinking about the right paper, contains quite a simplified view of this, and sometimes sometimes connections which they mention and draw on their pictures can be met, for example, only in 4 neurons out of 13, and they say it's like a system, periodical thing, which exists, which works, and so on. I need to compare and perform some analysis of this. I think it will be great if you will send me a link, and I will say, is it that paper, or I will send otherwise another one to you.
Yeah, just real quick, yeah, because I remember it was in this journal club, and Netta Cohen thought that this work was interesting, but then of course it's going to take me a minute for it to load up on this. So I'll show you in a minute the diagram I'm talking about. But why don't you keep going, and then when this loads, we can talk about it.
Okay. I will finish because it's the last slide.
Yeah. Go ahead. Yep.
Illustrates the connections between the same neurons, but the sources are, yes, that is that paper.
Yeah. Is that the one you were thinking about?
Yes.
Yeah. Okay. Good.
Okay.
That was just wanted
So
to.
the last slide, the last picture illustrates the difference between two alternative connectomes. Same cells on it show significantly different patterns of connections. There are two pairs of columns, left and right. So every part of the screen, one left and one right, contains two vertical lines, and you can see the difference pair one by one between pairs of the same neurons. So all of this illustrates the complexity of the task we are going to solve, and after we have seen all these examples, I think it will be good to discuss a strategy of what part of neural network can be selected as most perspective and valuable part for simulation in the most recent nearest time. So to select the nearest target for research.
Awesome.
That's the main thought about all this.
Great.
I hope this was at least somehow useful and important, but if not, it will be also important for me to know. Maybe I'm doing something wrong and useless, I'm not sure. So now I will be glad to hear comments about all this. Thanks for listening.
Thank you, Andre. One thing is, yeah, so it wasn't just that paper. Actually, there was another one too that goes a little bit further. This one is just sort of a simpler view of or a hypothesis of the circuit. But then this one here, which was also recommended by Netta Cohen, starts to it pulls out this level of detail for what they call these perimotor segments. It's in this Haspel and O'Donovan, and they have different ways of talking about like what they sort of think are these repeated circuits in here. But yeah, this is I think more what I was thinking about, that you might want to compare what they think they see in this structure to what you're seeing. Yeah.
This structure can be met, for example, in one segment, but there is no well, it is incorrect to reproduce it in complete. Each segment is individual, have individual connections.
Mm-hmm.
I tried to check the structure. It is not produced if we consider connectome data.
So you don't believe this picture?
Yes, I don't believe this picture.
Okay.
Especially near the head and near the tail.
Yeah. Well, I think even in the
there are additional mechanisms.
Yeah, I think in the paper.
And,
I think in the paper they are saying, like, they are making they point out that this has assumptions in it, and that it's not completely the whole picture. So yeah, I think even they're saying it breaks down, but yeah. But
And
anyway,
also each type of neurons has different number. One is 13, another is 11,
that's right.
6, 7, and so on.
That's right. Anyway, so cool. Yeah, it's good that you're looking into this stuff, because I don't think there's many people who are trying to compare this with what they really understand about this. I mean, it's a very complicated picture. So, but cool. Yeah, if you want to follow the reference to it, I pasted the URL in the chat there. Cool. All right. Other comments or questions for Andre from other folks?
No, I think it's interesting to start a conversation between the group that's doing the C302 stuff and Andre.
Huh?
Sorry.
Sorry?
Andre, I was saying, I was just proposing we should start talking, just doing meetings between you guys and, for example, Porek or Steven, also who's modeling the muscle cells and neuromuscular junctions. So to reuse as much as we can of that effort, because like this stuff of modeling the neurons is already happening, as you know, as part in other parts of the project. So I think it's time that we seem to be convergent, and that's great.
Yes.
So yeah, it's just a great opportunity to reuse some of the work that's been actually already been underway, right?
Okay, one more thought. I'm afraid that this data, all the facts, all the data about connectome and so on, which we have now, might be unavailable to build a really good working model. Possibly we need, using this data as something preliminary, to select some perspective part of the nervous system, and then interact with experimentalists in neuroscience to perform some experiment, possibly by means of optogenetics or some other technology, which allows to detect activity of neurons and even better for neuron processes to have a complete amount of data about how activity of some limited group of presynaptic neurons produces at next time activity of postsynaptic neuron or group of neurons. So in this case, this process can be simulated with some feedback between experiment, theory, and simulation, and we can be sure that the model finally works correctly, given this small amount of neurons, but works reliably. And otherwise, it seems almost close to impossible. So we can build these models, they can work somehow, but we cannot be sure about their relevance to the real ones. Am I right? Does it sound reasonable?
I think the trick is always that we know that there's all these gaps, and the trick is wearing our engineering hats and saying how many how many assumptions can we make, and how close can we get to what we understand to be the target, right? Because it's a multi-scale system, there's many different levels of abstraction that we can say it's either correct or not correct. So, I mean, something like the pharynx, for example, can we build a model that works either with the neurons or not with the neurons? Can we build a model that pumps at between 0.3 hertz and 5 hertz. You know, like there's always some sort of like macroscopic data that we have that we can build that starts to get closer, and then we can learn more about additional tests that our models have to pass, and then get closer. So I always think of it as just a matter of degree. It's not a question of like, can we either build it and verify it at all, or can we not build it and not verify it at all? It's, can we build something and verify it sort of, and then can we keep making it better over time? So part of it is just choosing a place to start that we feel comfortable with for, you know, and we've done it for a few different circuits. So we've done single muscle cell, put some work into the synapse, we've got some work on the motor neurons, we've got small circuits that we started to look at least build up the infrastructure for. You know, we could do more on the pharyngeal neurons if that's, and the muscles there, if that's a good place to start for your investigation as well. We have some of these tools now that we can start to build these models up and ask more detailed questions about them. So, but yeah, it's a question of what do you think is the most interesting place for your stuff to begin, and then, you know, maybe we can all kind of converge in a single space. That's how I think about it. Other questions or comments, anybody? Okay. Well, so for next time, I think I have not yet gone in a given section. So I will volunteer to talk next time about OpenWorm, muscle model, one of those two things, one of those two topics. Also be good to move that stuff forward. So I will volunteer for that at the next meeting. And now let's go round robin and talk about what's new. So Michael, do you? So by the way, thank you, Andre. That was cool. It's good that we keep thinking about that stuff. So Michael, you want to go ahead?
That was the most complicated Excel spreadsheet I've ever seen with those wirings. So hats off to you there, Andre. That's super cool. Yeah, so I can give a very quick update about movement validation. We had our monthly meeting on the 15th of April, and it was attended by myself and Jim, and potentially two potential new contributors, David Clark and Aidan Rock from Edinburgh University. So Aidan is working with Balash this summer to help his work along, and hopefully that will work well with potentially making contributions into the movement validation repository. So he's looking to adapt the motif analysis of Andre Brown and incorporate that into our code base. Because right now, of course, we just have the Yamini's features translated, but it'd be cool to have somebody else's, potentially Andre Brown's motif analysis. So he's working on that, or will be as soon as his exams are done. And in fact, speaking of Andre Brown, I received an email today from Kezi Lee, who gave us an update about his work on the computer vision algorithm. So he's done a whole bunch of work on that, and that's up on a GitHub repository that I've linked to in the meeting notes here. And so I'll be having a conversation with him about how to integrate that into movement validation so that we can hook up this pipeline, because that's really the last major piece of the pipeline, other than, of course, the videos at the beginning of the thing. But of course, I heard about our good AWS news that we've heard from them, so hopefully that will initiate the whole pipeline, and then we can process it with this computer vision algorithm and then go from there. So that's good news. And then finally, I can just a short update on myself. I've been continuing to work on the serialization of our objects so that there can be information transmission from Geppetto and the rest of OpenWorm into movement validation. So still continuing to work on that and get that basic worm minimal format serialization working. So that's movement validation.
Cool. Michael, I feel like you and I have switched outfits for the purposes of this meeting. You're usually like all wearing a tie and stuff, and now you're more open collar, and usually vice versa. So.
Well, you're on the East Coast, so you gotta dress up, right? On the West Coast, they'd say you're working at J.C. Penney or something, but on the East Coast, you gotta look good.
There you go.
Even as a software guy.
There you go. Awesome. Cool. Thank you for that. Sounds like good stuff's happening. All right. Let's see who's next on our list. I think Matteo has given a talk. Is in Germany for giving a talk actually on OpenWorm and Geppetto right now. So unfortunately, he's also absent. But maybe Giovanni can provide some updates in the general space via the WormSim stuff.
Yeah. Okay, so yeah, there's a lot of stuff going on with Geppetto that I'm not going to go into too much detail because most of it is major refactorings to add the dashboard, and there's a new domain model being refactored into the engine. So that's pretty much what's going on, and there's like 4 or 5 people actively working on branches to pretty much gutting the engine, and it's going to go out in the next release. But yeah, so that's driven by the fact that now the simulations will be persisted. So there's going to be a dashboard in Geppetto with your simulations, basically. And this is driving the fact that there's a lot of changes, and the simulation files are changing. Now they're going to be what currently is the XML simulation file is going to become just a model description, and the simulation file will contain a pointer to this model as well. And the simulators are moving out of the current simulation file. So there's a lot of stuff going on. We're separating the models from the simulators and persisting the simulations, and then now they're going to be called Geppetto projects. They show the simulation and so forth. So that's what's happening. There's going to be a dashboard, and this is causing a lot of changes. Plus, there's a lot of stuff happening in terms of external simulators. We currently are introducing the ability to talk to any external simulator that is wrapped via command line, pretty much. You can talk to anything that sits on your server via a Geppetto bundle that gives the instructions to this third-party kind of external simulator, and then it pretty much configurable where the results are going to get dumped, and Geppetto imports this as a Geppetto recording. So that's very much what's happening. Dashboard and external simulators, and a lot of refactoring going on around that stuff. So that's what Matteo's update would have been, but in more detail. Now I'm gonna go to my stuff, which is more in terms of the WormSim, and I got the chance to do some coding myself, other than just coordinate everybody on that branch of the OpenWorm project. So what's happening right now is that in the last couple of weeks, I generated HDF5 recordings from the SPH process data, and this data is going to be read through Geppetto, but also a little refactoring on the back end is required to read the recordings in such a way that the stuff ends up where it should in the runtime tree. So I don't want to go into too much detail because that's really like implementation details. But yeah, so the HDF5 recordings for the SPH process data have been produced. So we have a bunch of recordings files in HDF5 formats for what it's going to be the WormSim. So the data for the mechanical simulation is going to come from these recordings, and then yes, there's some refactoring of the backend on the Geppetto side of things to enable for streaming some of the stuff, because with these recordings we are introducing into Geppetto the ability to animate 3D scenes based on stuff that's in the recording file. So this wasn't possible before, so there's a bit of refactoring going on to add this feature. So that, for example, in a recording, not only there are simulation variables, like state variables, values coming, so for example, for a muscle cell, the activation signals, but also there's going to be information for how the physical representation or the mechanical representation of the muscle cell changes its shape in time. So that is a visual transformation that wasn't there before, and we're adding that feature.
So I just wanted to point out to folks that if you remember this diagram from before, that the stuff that he's talking about just sort of still fits into this bigger picture here, where basically, yeah, stuff is coming. He's working on this bit, right? Coming out of cybernetic. There's the particle-based simulation. We're extracting the skeleton, turning that into the skin mesh animation, and then he's reading in this content. And these are, which one is the HDF5? Is it?
That's it. That pretty much there should be an arrow from that to Geppetto, because the HDF5 is that pretty much. The skin mesh animation, not pretty much. The dead matrices for the visual transformations are going into an HDF5 file together with the muscle activation signals.
Yeah. Oh, so their muscle activation signals are also kind of coming in as well?
Yeah. Together with the transfer. So basically, the SPH recording that we have in HDF5 has the skin mesh animation, so the matrices for describing the animation, plus the muscle activation signals. And this stuff gets fed into Geppetto. The worm sim is something that, in a sense, reuses Geppetto. So that arrow is not necessarily wrong, but I think that's pretty much why it's there.
Okay, cool.
Yeah, so that's pretty much the pipeline that we're building. And there's a bunch of people working on this. So Matteo, for example, is helping with design discussions, and just there's a lot of this stuff. There's a lot of overlap with Geppetto refactorings and stuff, so we're kind of working together on that. Plus, Matteo is also helping with UI and UX, kind of more detailed mockups compared to the ones we had on the use cases document that we drafted 6 months ago. So now we're implementing those screens to kind of fill in the gaps. And then Sergey is also helping, and he completed, for example, a looping simulation from the SPH particle-based simulation, including the activation signals, so that we could go through that pipeline that Stephen was showing. And Martin is the guy who worked on the skeleton extraction from the particle-based simulation, and that stuff is done. Also, Sergey is helping us with mapping the muscle cells in the model to the muscle cells in the anatomy, so that we have the names for each. So that's, we were talking about this last week, this current week, so that's going on. And then Mihai is working on adding functionality in the Geppetto front end to play the animation that's coming from the SPH recording. Plus is also working on implementing the worm sim UI UX mockups that Matteo is putting together. And last but not least, Balázs is helping us out with tutorials, and I'm also working on those tutorials myself. So yeah, that's pretty much everything. Questions? I'm sure there are a lot of things that I licked.
Did you want to show the mock-ups that Matteo posted on GitHub?
Yeah, they're in a card. I'll just bring them up. It's probably gives more of an idea of what I'm talking about.
I saw this fly by. The mock-ups are to make what you've seen already from Giovanni last time we met, a little prettier. Just to sort of make it even shinier.
I'm bringing them up. Share my screen. So this is the latest mockups of what the worm sim customization, create your worm before you go into the simulation environment, you pretty much, there's going to be a welcome page, and there's going to be a worm, and you can read this in detail. And then it zooms into the worm, and you can pick a color for the cuticle. That's going to also retain this color in Geppetto. And then you can give it a name. And Matteo decided to call it Spark, and it's blue. And then after you finish this phase, pretty much this is the mockups I can show, because the other ones is working on them. But after this, you're going to end up on the main page where we have a mini worm browser. I'm going to show you. I probably still have I need to check if the instance is live right now. But I won't show you what happens after this, but the screen will be reworked. So maybe, yeah, it's live. I need to share another thing. So after those mockups that I have shown you, we're going to end up on this other page that I'm sharing now. Right? So there is a mini worm browser. This is going to change into a slider, and you can click Explore a crawler worm or Explore the nervous system. This is the name. I just called it Yellow Worm. That's my worm. And when you click these buttons, it opens the simulation environment, which is pretty much a customized version of Geppetto for the worm sim, with custom components such as the logo and on button to come back to this page. So, but this page that I'm showing now is going to change. Matteo is currently kind of beautifying it in the mockups, and Mihai is implementing that stuff. So yeah, that's pretty much what's going on. So when you click on this Explore the Chlorine Worm, it's going to go and replay the SPH recording. Then you go and click on Explore the Nervous System. It's going to go and play the C302 Brain of the Worm simulation. And it's not just going to play those, it's going to be kind of more educational than that, because there's going to be tutorials with little balloons telling you, if you click this, this is what it means, try and do this, try and do that, try and plot this neuron's activity. So it's going to be interactive in the sense that the user is guided through through whatever is going on, because if you're not familiar with that stuff, the purpose of this is to expose it to kind of a wider audience who might want to learn about biology a little bit more, and the stuff that we're showing is sometimes not self-explanatory inherently. But yeah, so that's pretty much everything from me. Hopefully it was.
Yeah, awesome. Very good. Any questions for Giovanni? Okay. Sergey?
I also worked with Andrei on exploring neuron system of C. elegans, and I made myself my part. By the way, I started to work on interface between a neuron and cybernetic, and have some preliminary version of it. It now can work I just made script which can work with neuron from Python, and we know that cybernetic can work with Python, and potentially it will be simple to connect each other. Actually, this code it's now not in GitHub, and I plan to add it on GitHub soon. I can show you what I have for now.
Yeah.
Okay. Unfortunately, I have a problem in on my home PC with some library. It couldn't be found, and by Neuron doesn't want work. So I'll show you what I have. Just a moment. So I have here some part of model sub-circuit of some neurons. This model was created by Andrei.
Hang on, does anybody else, are folks seeing it? Because it's like...
Oh.
Black screen.
Sorry.
Yes.
Oh, I don't know how to fix it. Actually, there are some OpenGL window. Maybe there are problem.
Try sharing the whole desktop, maybe, instead of just one window.
Try.
Yeah, well, sometimes the video card doesn't send it to the
yeah.
work.
Do you see this?
Yeah, yeah, no, yeah.
So it's a window opening when I start this script. Neuron load. I mean Pine neuron, which is interface between neuron and Python, loads model of which was made by Andrei some times ago, and this shows this OpenGL window shows this model and some neurons. You can see this here. So, and actually it should start simulation on neuron, and we should see changing of brightness of some segments of these neurons, which is indicated changing of voltage in this segment. But I can't show you here because I have a problem with neuron. I don't know what's happening here. But on my workstation machine it works fine. So next.
Oh, sorry.
Next step, let's connect this script with Cybernetic and made some useful additional features in this interface. So, Andrei, what do you want?
Okay. The main idea here for creating this was to make an alternative visualizer for Neuron. Because original one has quite limited functions. For example, we cannot use different colors to display different types of neurons, and when we plan to simulate something quite complex, like, for example, a fragment or a whole locomotory system, which includes a few dozens neurons, we need to color different groups of neurons with different color. So also we plan to add some functionality, like, for example, to conveniently activate selected neuron with some mouse command and so on, which is also unavailable in original Neuron. So this software is intended for improving its usability, convenience, functionality in application to our purposes. It also will be able to connect to cybernetic, as Sergey mentioned. So we hope that in one window we will be able to see crawling worm, and in another it's a fragment of its nervous system with visualization of its activity of neurons, compartments, even working synapses, as we hope, because there is no way to display synapse to make it visible and to distinguish, for example, between synapse and electric junction in original neuron, taking into account that they are supported at the level of the model, but it's hard to display them in visual scene. That's it. So thanks, Sergey, for talking about this.
Cool.
It's a very good start.
I want to see a neuron changing color, and I want to see a muscle going, like changing its shape within SPH. If you can do that, it will be huge. We can connect those two together.
Next step.
It's awesome. Cool. All right, Andre, did you have any other stuff other than what you presented for an update? And that's pretty much at the beginning, pretty much everything that's new.
Well, now we are thinking about Sergey will continue working on this software, and I will in parallel produce some network which will work inside it.
Cool.
I was trying to choose the best variant, and my search produced this amount of data about which I told today, because sometimes I met very interesting things and I started to collect them. So that was the result. But I'm still not sure about what small neural network will be the first to implement as a model.
Hey, Andre, can you share the document that you were showing somewhere?
Yes, I think I will provide it with links to all the facts. And then,
Okay.
What is a good place to put it? Just to share with Google Docs?
Yeah.
Just throw it into the Google Docs.
Okay.
Yeah. That'd be awesome.
Okay, I hope tomorrow I will do it.
We have a folder called biological details. I think maybe be good under there. Maybe start a folder under there as well. Look for that one. That's where we put some of the other past stuff about muscles as well. So that'd be very good. Okay, and so we've come back around. So Gopal, who's been observing here today. I think Michael just joined after Gopal, but if you were watching on YouTube before, you probably heard that he's a new contributor looking at some of the OpenWorm and muscle model stuff. So any comments or updates or things that why don't you tell us like what you've at least read about the project and what you've been able to get into, if anything, even if it's just like very minor or just like stuff you've read. This doesn't have to be huge, but just some kind of update from you.
Yeah, sure. I mean, not much, to be honest. I was actually having some strange installation problems in getting the muscle model running on my computer, which I haven't managed to resolve yet. I think it may have to do with macOS 10.10, and so that was sort of my roadblock, and before I got caught up with work-related things. So, I mean, I guess moving forward for me, I'm going to be starting school, medical school, in a couple of months, and so something I'd like to figure out, or things I can work on that are maybe not so time sensitive. And so, I've just been kind of browsing through Muscle Model and PyOpenWorm. One thing I've thought about would be, doing a lot of unit testing. It's a good way to get a broad overview of what's going on. So, any suggestions would definitely be welcome. I'm eager to jump in on anything, really.
Okay. Yeah, I think the best thing is for you and I to get a time slot and talk, and to have some code as well. I'm just remembering that we can meet up in person here, but also maybe to sit down with a computer and see if we can resolve installation issues, and I can show you more stuff. So let's get some sort of time for that.
That's great.
Cool. All right, guys. Well, we are right at the top of the hour. Anybody have any other additional comments or anything to make here for the good of the order before we wrap up? All right. Awesome. Well, I think next time we'll get Matteo back here and he'll update us on Geppetto. We haven't seen him in a while, but I know that he's been busy and he misses us. As well, some other faces, Oregon and Balash. I think we'll make sure to get him back here next time as well. But thank you, everybody, for the progress. Things are looking good. And I will catch you again at the next general meeting.
Thank you.
Thank you.
Thanks, guys. See you.
Thanks, everyone.
Bye.
Bye-bye.
Bye.